So what we're going to talk about is relapsed and refractory CLL. And this slide's actually about 4 or 5 years old. And what it shows is really the average number of treatments a patient would have gone through in the pre-era of the new molecules. 100% of patients diagnosed, 1/3 don't need treatment over time, 2/3 do, 67. First line, second line, third line, fourth line. 6% we're getting to fourth line. With the new agents now, we're going to see a lot of patients go through 5, 6, 7 different lines of therapies during their journey through their illness. So part of what we've got to think about when we're thinking of relapse and refractory, the second line therapy, is actually how does that complement the first? And are we actually ruining our chances of further therapy later on? And we have to start thinking about the long-term management of patients, not about this round of treatment in the way we have historically. So just a bit of terminology for you. Refractory either means your treatment didn't work at all or you've relapsed within 6 months. And the general rule is try something new. There's no point in giving the same treatment again. With relapse, means your disease coming back at some later point. And we call it high-risk relapse if it's come back within 24 months. And rather arbitrarily, again, the rule is try something new. If it's over 24 months, the dogma has been it's not unreasonable to give the same treatment you had the first time. And that's been the historical dogma, which may or may not be correct in the new era. But to go back to our old friend of fludarabine, cyclophosphamide, rituximab, this is the MD Anderson data, the 300 patients published 2004. 97 out of 300 still in ongoing remission. So what a very effective treatment it is. But what they looked is at their relapse and refractory patients and what actually happened to them. At the MD Anderson, very much clinicians do what they think is best. So there's no set rule of how patients are treated. Overall, after FCR, when you had your second treatment, whatever you had, the average duration of response second time round, survival, is still 4 years. So even after your disease has come back from FCR, and on average that's 4 to 5 years, on average people survive in another 4 years. And this is very much data from the pre-small molecule era. So patients still did well. When you actually look though, the elderly group on the right, left line, the old pointer's playing up. If you're over 75, you did phenomenally badly if you relapse from your FCR. Because there's probably no way they can give you the FCR a second time at 75. They did very, very badly. That's a real unmet need for what do you do with what are, in fact, the majority of our patients over 75. If you look here, if you look on this one, however, basically, if you'd relapsed within 1 year of your FCR, you did phenomenally badly come what may, whatever you gave. If you relapsed within 3 years, again, you did less well. The longer you relapse, longer your periods of remission from the FCR, the better you did. And that's where this dogma came out that around 2 years, there's no point in giving the same thing. You've given FCR, there's no point in doing it again if you've only got 2 years. The benefits versus the toxicity are just not there. Because if you give the same treatment again, you're always getting your first duration of your remission minus something. You never do longer and you never do as well. So what did they do at MD Anderson? And this yellow line here, the top one is lenalidomide, but it's only 12 patients. Although it looks impressive, it wasn't many patients, so I'd ignore that. But in fact, oops-a-daisy. Most of them, a lot of them got FCR again, that's the blue line. There are other treatments given rituximab-based, bendamustine-based, but the best treatment after FCR if you relapsed was actually FCR again, if you could tolerate it. And as we all know, a lot of patients can't tolerate 6 FCRs first time round due to the toxicity of it. But they're tough folk in Texas, as we know, because they got a lot of them for it a second time. And if you can, it may be good for you. But if you actually look at survival after the salvage, it's phenomenally low for an awful lot of patients if you'd relapsed within 3 years of your first FCR. Whatever you did, you were going to do badly. Relapsed after 3 years, there's the FCR line, you do better. And that's why that cut-off of 2-3 years is almost a biological marker of you and your disease. Let's say this was the pre-small molecule inhibitor time. So the standard dogma was if you relapsed after FCR, you could actually give it again. Maybe have to go a bit lighter with it because of the cytopenic effects, bone marrow toxicity. Maybe you'd move to something like bendamustine rituximab, a bit less toxic. Well, there's a whole host of things you could have given. Obviously, 17p, you don't treat with chemotherapy, you shouldn't. So that was the basic dogma up to the last 2-3 years. And then what we've had is a whole revolution, a bit like, say, London buses. You've never had one and then 4 come along all at once. And all of a sudden, we got these fabulous drugs, ABT19, Venetoclax, Ibrutinib and Idelalisib to start with, to actually change the paradigm we've talked about. And what CLL has done, what people have done for the last 20 years, is sit in their laboratory rather sadly and work out what keeps the cells alive. And this thing called the B-cell receptor on the surface of the cell we now know has been pivotal in keeping these cells alive. And it actually sends signals to itself to keep itself growing. And there's a molecule here, BTK, and we found that by inhibiting that molecule, the CLL cells died. There's other BTK inhibitors coming along, which is good news, because if you have a monopoly, it's never quite as cheap as it might be. There's Idelalisib PI3 kinase. There's another, There's another one coming along as well. And this drug, Entospletinib, is a Syk inhibitor, and that's in clinical trials as well. So you've got 3 different ways of switching the cells off. And one of the interesting things about that is we thought if you're resistant to one, you may be resistant to another. And that's actually not proven to be the case. If you're resistant to one, you can cross over to the other and may still get response. And that's been very exciting news in the last year or so. So moving on to the pivotal study with Idelalisib. Idelalisib rituximab versus rituximab alone. I actually refused to do this study on ethical grounds, because in the UK, rituximab monotherapy is not a recognised treatment. And therefore, I refused to do it. But I found myself in the minority in the UK. Everyone else seemed to do it. But I didn't. For once, I had the high moral ground. What a rarity. And anyway, moving on. The very interesting thing about this group, if they had a lot of patients, was 17p deletion. 42% of patients had a 17p abnormality. In that age again, 71 years of age. Real people with real problems. The number of prior therapists, 3 therapies, 1 to 12. I couldn't think of 12 different therapies in CLL so God knows what these patients have been getting, but who cares? It kept them going. They got to the trial. Excellent. And if you actually did progression-free survival, you did significantly better with Idelalisib than you did with rituximab alone. It's a relatively short duration of follow-up here. We're only just over 1 year, and you're getting down to almost half the patients, the disease is waking up again within 1 year. If you actually look at overall survival, you get a significant benefit from it. And they salvaged some of the rituximab patients by crossing them over to the Idelalisib. When you look at the toxicity of these drugs, the very interesting thing is it's not a chemotherapy. And therefore, you're not getting that neutropenia-type problems, that nausea, vomiting, diarrhea-type problems on the whole with these agents. But what we did get with Idelalisib is colitis, which is usually occurs 4 months or so onwards. The patient's smashing, the disease has gone away, and then they get colitis. And you also got pneumonitis, or pneumonia if they called it at the time. And what we now think is it's affecting your immune system itself, so you'll get an auto-reaction to the bowel and the lung with these. We also know that a lot of these patients turned out to have pneumocystis infections which wasn't appreciated at the time of the study. The other licensing study came along for relapsed and refractory with the Ibrutinib versus ofatumumab. We did do this study. And cutting straight to the chase, progression-free survival, really quite amazing, 60% in a phenomenally heavily pre-treated group of patients. Overall survival, again, once they managed to cross them over, the line flattened out. And the really amazing thing with these agents is it doesn't matter if you've had chemotherapy before, you almost respond as well as you've had chemotherapy before. Whereas before chemotherapy, following chemotherapy, you never did as well. So you'll get a longer duration of response than your previous chemotherapy. So if that was 3 years, you'll be getting longer. If that was 1 year, you'll be getting longer. That was never the case when we had chemotherapy alone. Again, the great thing, whatever your prognostic markers, take your pick, they all did better with Ibrutinib, including the 17p. The toxicity, again, very low indeed. Diarrhea is down in the 1-3%. Infections is down in the 1-3%. So Ibrutinib looks to be as effective as idelalisib. It's hard to tell if it's better or not. There's been no direct comparison. And I think most people think the idelalisib study had more ill patients in it, certainly more 17p, and therefore it's hard to directly compare. But toxicity-wise, it certainly looks at Ibrutinib as the less toxic of the two agents. But it doesn't matter. If you're idelalisib and you don't get the toxicity, it's a wonderful drug. And then you come to what we call the real world. Well, in our case, we talk about the UK. There's nothing real about that. It's a bit surreal at the moment in the UK. And this was published only a month ago. And what Janssen did, quite amazingly, they opened a named patient program. One of the great things of going through NICE, if you can drag it out, a lot of companies give you the drugs free. In the space of 6 months, they managed to find 300 patients in the UK to give Ibrutinib to. And George Follows from Cambridge, with Janssen, decided they would actually have a look at how these patients did. And then you get a slightly different story. If you look at the patients here on Ibrutinib, half of them have stopped it within 2 years. Overall survival is down here about 60% at 2 years. Nowhere near as good as was seen in the studies. When you look as to why that is, in the study, your performance status, you had to be 2 or below. If you look in the UK, those people with 2 or more comorbidities did significantly worse, even with Ibrutinib. And the same with overall survival. Same drug, but there's the people with the comorbidities. But one thing very interesting that came out of this study is there probably was a learning period for the doctors actually doing the study. And one of the problems with these new agents, anyone can hand out the prescription. The boot is not toxic. Just hand it out and let the patient get on with it. It's so simple. If you had what you thought may be a toxicity, if you stopped the drug, you are much more likely to die than if you reduced the dose down. So the message from this, if you think you have got toxicity from Ibrutinib, is drop the dose, don't stop the drug, unless you absolutely have to. And so in the UK, we think probably a lot of doctors, because we didn't know this, and certainly Janssen hadn't told us, a lot of doctors were learning how to use the drug with their patients for the first time. So in the opening of this whole program, everyone could use it, not just those who had done the studies before. So there's a learning period for doctors as well as for patients. Just to move on a bit, it's a Helios study, which is bendamustine rituximab with Ibrutinib compared to bendamustine rituximab alone. You do significantly better with Ibrutinib added to bendamustine rituximab. If you look at this study on the right, the Jamberger study, this is Ibrutinib rituximab. If you look actually at the progression-free survival here, it's not significantly different to the eye for the two regimens. So you have to ask yourself, are you really getting much out of the bendamustine? You may be, because you look at actually overall survival, the bendamustine R group do slightly better. But I think we're going to have to start questioning the role of chemotherapy in some of these relapse regimens. Do they really add benefit to it? And another study, which I won't show you, the Gilead 1115, bendamustine rituximab idelalisib versus bendamustine rituximab. The progression-free survival looks phenomenally similar, idelalisib rituximab, as it does with bendamustine added. Moving on. What is apoptosis? You know, things just die all the time, don't they? But actually they don't. Apoptosis is a deliberate act by the cell to die. And let me ask you one silly question. Why does your hair keep growing, but your nose hopefully doesn't? Come on.
My hair falls out.
Pardon?
My hair falls out.
Yeah, well, we can't, some things we can't cure. Let's get, your hair keeps growing because your hair follicle just produces the cells whether you need them or not. Your nose doesn't keep growing because you eventually reach a state you only produce one more cell on average when one cell dies. And if you take your blood count, those normal ranges of neutrophils and platelets, there's conditions called myeloproliferative conditions where that break to switch off your platelet count and stop you producing too much because that deleterious fails. So we have the whole body system is designed, you get what you need of a number, and if you don't need it, you kill it. And the cell does that itself, and that's called apoptosis. And we now know all the proteins which regulate apoptosis. And if these ones up here, BCL2, which is a very important one. And what we now have is this drug available to us called ABT199 or Venetoclax, which actually directly blocks BCL2 and makes the cell say, I've had enough here, I think I'll just croak. And the cell dies itself. This is the very earliest report of Venetoclax in patients who had all sorts of other treatments. It's an oral drug. We saw tumor lysis with it, and tumor lysis actually means you're killing the cells more effectively than anything else. And for an oral agent, it's not a chemotherapy to do that. You can get it with FCR, but actually it's really quite amazing. You don't see that with Ibrutinib or idelalisib, so this stuff really kills the cells. Just to move on quickly, what about allogeneic transplant? We really reserve that for the relapsed refractory high-risk group 17P. But I've got a lot of patients who I've offered transplant to, and they've opted to stay on their small molecule inhibitors. I've had only one out of about 10 or so I've offered transplant to. They would rather take their chance with the small molecule inhibitors than allografting. So it's given us a bit of dilemma with these patients. There are over 1,000 registered studies in the world of treatment for CLL. And this is why I'm quite convinced we're going to get the answer. There's so much money going into it, so much effort going into it worldwide, I'm quite convinced. And just randomly pick out, lenalidomide's been around a while, but we haven't really found a use for it, but immunomodulation is coming back. And the studies maintenance only with rituximab, chlorambucil, BTK inhibitors, with Ibrutinib, nivolumab, is a slightly different. It's a monoclonal antibody, stimulates your immune system, very novel. There's entospletinib, stick inhibitors coming along, all sorts of things. And Venetoclax, what do you want it with? Cornflakes, Rice Krispies, obinutuzumab, rituximab, Ibrutinib, bendamustine rituximab, and I could have gone on and on and on. There are loads of studies out there. What we're going to run out is patients to do it. If these new agents keep working so well for so long, where are these patients going to come from to go into the studies? That'd be a lovely problem to have. Vaccines are still around. There are vaccine studies going on for CLL, which are very interesting. And I've got down here autologous T cell, right in the bottom left-hand corner, CAR T therapy. And this has been with us 5-6 years, really in experimental centres. And we know from allogeneic transplant, a person's immune system can kill their CLL. That's what you're doing with the transplant. You're giving them a brand new immune system. And with CAR T cells, they take your own cells out of you. They manipulate it in the lab to direct them to kill your own cells, and they give it you back. And when it first came out, people were saying, what, $500,000 for that? Who the hell's going to pay for that? Well, actually, you're not going to be far off that with 3-4 years of Ibrutinib therapy. So CAR-T is improving as a technology, and I think will become affordable in the big scheme of things. And I think has a real hope to actually get that C word into chronic lymphocytic leukemia. So at the moment, just to the NCCN guidelines, you can basically, all sorts of things are recommended. You know, you can almost come back to what's not on the list in terms of what they recommend you can do. For the 17p, it is slightly different. There are only certain drugs that work: Ibrutinib, Venetoclax, lenalidomide, and steroids. Although ofatumumab is on the list, it's really a very poor agent. I'm not sure OFAR has much to offer either. Endless choice, but you're back to, you wouldn't need all that choice if we had one that did the job for everyone. So in conclusion, many therapeutic options. The scheduling, it does depend on what your previous therapy was. It very much depends on your duration of response. The general trend is away from chemotherapy a second time. If you can possibly avoid it, the toxicities are still there. It also depends on the cost of the drugs and the regimens. And really, you do need to sit down, talk to your doctor as an individual, talk about you as a person, and come up with the best plan for you. There's lots of choice. Thank you very much.
For Dr. Chris Fegan, you said that there's over 1,000 different trials, or over 1,000 trials. The first question is, are they all different, or do they look at different, do they have different medicine, etc.? And the second question is, are they looking for one particular cure for everybody with CLL, or are they looking multiple treatments for different groups?
The answer to that is they'd love to have a cure for every single patient. That's called ka-ching in most people's minds, but the truth of the matter is they're really trying to look at subsets. And one of the things going forward with clinical trials is actually what is the unmet need. As Kostas said, you know, we need to think of a new way of treating the not one patients, because the FCR is not the answer. So a lot of these trials are actually more stratified patients, identifying smaller groups of patients, and that's why you need international collaboration, because one centre or one country may just not have enough of these sort of patients, and that's why you have to go worldwide. Unfortunately, that makes the study a lot more expensive. But one of the problems as well at the end of the day is we as doctors are not usually developing the drugs. They are developed by commercial entities. And how we're allowed to do the study in lots of ways is also dictated to us by the commercial studies. And if you're dealing now with drugs which are very different, and if you take BTK inhibitors, there's now 3 companies producing them, they will only allow you to put it together with other people's drugs, other companies, and some companies like each other and some companies don't like each other, and therefore you can't necessarily do the study you want. In the old days, you used to be able to do the study you want because the drugs were fairly cheap and the government would give you the money. So FCR versus FC, you really didn't need much from Roche's approval to do that study. But now these agents are, you know, $100,000 per patient for a national body to fund the study you'd really want to do, or the patients would really want to do, is very difficult. So we are a bit hamstrung at times. With what we'd like to do, and we are a bit driven by the commercial sector, but then thank God they're doing the work, because otherwise we wouldn't have the new medications coming through. I'll just say one thing to Brian. One of the interesting things about the Rituximab versus Rituximab placebo study, which I declined to do on ethical grounds, and I asked Gilead, how did you ever end up with Rituximab as a single arm? And they say they discussed it with the FDA, of what would the FDA accept as a good control arm. And the same is true of the resonated Ibrutinib versus ofatumumab. So since some of the regulators need to actually look at themselves as well, because I don't think it was a fair comparison, and Kostas mentioned yesterday the dose of chlorambucil, we were all using the dose in these studies, I say we were all using the dose in these studies is low. So why are the regulators accepting that as an acceptable alternative? And I think they need to raise the bar, because they've got the power of the FDA as patient advocates.